Using flow cytometry to speed determination of eukaryotic genome sizes and cell type-specific gene expression, presented by Genetic Engineering and Biotechnology News. Thanks for joining us today. I'm Tamlin Oliver, Managing Editor of GEN, and I'll be the moderator for this webinar. Flow cytometry provides a convenient and accessible method for analysis of plant genome sizes and for other comparative studies. Application of this technique to plants has been limited, however, due in part to the need for sample preparation that yields particle suspensions without cell perturbation, the limited dynamic range of conventional flow cytometers, and the affordability and complexity of most flow cytometry platforms. The three speakers today, David Galbraith, Chuck Cannon, and Claire Rogers, will address these concerns and share their solutions in a minute. Before we get started, though, I want to encourage you to submit questions for our panelists at any time during the presentations. We'll answer as many questions as we can after the final presentation. What you need to do is type your question into the Ask a Question box on the lower left of your screen and hit Submit. Our first speaker, David Galbraith, the professor of the Bio5 Institute and School of Plant Sciences at the University of Arizona, will talk about the technical aspects of flow analysis and sorting in plants, including a chopping method that facilitates genome size measurements, characterization of natural and agricultural populations, and unsuspected phenomenon and molecular and cellular biology of the nucleus. David, we're ready when you are.
Okay, thank you, Tamlin. My duty now is to give you the technical aspects of flow analysis and sorting, and particularly as they apply to higher plants. And in the next slide, we'll just see a schematic of a flow cytometer. The center of a flow cytometer is the flow cell, out of which appear a stream of cells or particles that are going to be analyzed. And these intersect a laser beam and absorb light and emit this in the form of light scatter or fluorescence signals, which are detected by a series of photomultiplier tubes. The center of the flow cytometer, the flow cell itself, has a flow tip, which typically has a diameter of 70 microns or thereabouts. So in the next slide, you'll see the simple issue with dealing with plants, which are, of course, multicellular organisms, is how to fit a plant through a flow tip. You see three examples here of a Arabidopsis thaliana, which is the model plant that's used for many molecular studies. In the next slide, you'll see one approach can be to make protoplasts. That is, we digest away the cell walls and we release single cells. And in the cases I show here, we have leaves or we have cell cultures. And in both cases, you can make these suspensions of single cells. These, of course, will fit through a flow cytometer. But when we started doing this work, we were interested in measuring the genome sizes of the individual plants. And if you go to the next slide, you'll see a DNA flow histogram in which we've taken some tobacco leaf protoplasts, the single cells, and we've fixed them. And then we've stained them with the DNA fluorochrome and run them through a flow cytometer. And you see a peak, a rather broad peak around channel 150, which is representative of the fluorescence produced by the fixed tobacco protoplasts. The DNA content of these tobacco protoplasts is about 3 or 4 times that of chicken red blood cells, which you can also see in this histogram. But the important thing is that the CV of the peak is rather poor. It's 7 to 10%. The reason for this is that the protoplast is rather large. It's about 40 microns in diameter, and this means it's illuminated unevenly within the flow stream. Now, if you go to the next slide, you'll see that if you break open the tobacco protoplast without fixation, and you can do this simply by adding detergent to the staining medium, you end up with a coefficient of variation of the DNA peak of about 3.5%, which is pretty reasonable. So this represents the fluorescence being emitted by the tobacco nucleus itself. And the improved CV is simply because the nucleus is more evenly illuminated within the center of the stream as it goes through the laser beam. So you might say then, okay, we'll just make protoplasts from all plants, and we can run them through the flow cytometer, and we can measure them after breaking open the protoplasts to measure the DNA contents. The problem with this, as you'll see in the next slide, is that protoplasts are not really particularly easy to prepare. And in fact, some species and organs cannot be made into protoplasts. And we confronted that problem, and we thought to ourselves how to solve it, and then realized that it would be very easy simply to release the nuclei from the intact plant rather than making protoplasts. And we devised a method of doing this, which was a high technology approach, taking a single-edged razor blade and chopping up the plant material. And this makes a homogenate. As you'll see in the next slide, the method, which we published way back in 1983, is simply a 6-step procedure. You take the tissue of interest, could be leaves, roots, seeds, shoots, any part of the plant really, place it into a Petri dish, add the chopping medium, chop the tissue with a single-edged razor blade for about a minute, and then filter the homogenate that you made through a nylon mesh. And this just removes all of the chunks of tissue that would otherwise plug up the flow cell tip. So the porosity of the nylon mesh, 15 to 50 microns as needed, and then you add the DNA fluorochrome to the appropriate concentration and measure the fluorescence emission of the nuclei within these homogenates using flow cytometry. The next slide in panel A, you see a DNA flow histogram of a chopped tobacco leaf, and you can see the resolution of the G1 peak, that is the 2C DNA content of the cells as released, as the nuclei are released from the leaf tissues. The CV is 3.5%, which is really very nice, and you can see the cell cycle, G1S and G2 peaks on this linear histogram, and this is a very acceptable DNA histogram profile. And we went on to measure other plants. You can simply go into the greenhouse and take as many plants as you want, chop them up, and the panel B shows chopped Arabidopsis leaf. And when we did this work, we were interested to find out that instead of having a single G1 peak with a 2C DNA fluorescence, you can see 2C, 4C, and 8C peaks. And, of course, these are all lower in DNA content than the chicken red blood cells because the genome size of Arabidopsis is much smaller than that of chicken red blood cells. And then the little inset panel shows on a log scale that the peaks are evenly spaced, as you'd expect if you were to see endoreduplication within the somatic cells. So this was unsuspected, but quite interesting. The next slide illustrates the applications of the chopping technology. This is used worldwide. It's used for essentially all plant species and tissue organ types, for making genome size measurements and also for looking at ploidy levels and for looking at the cell cycle. It's used for characterizing natural and agricultural populations. Ploidy screening is quite important in agriculture. It's important in breeding certain types of fruit crops. For example, watermelons, triploid watermelons are seedless, and ploidy screening for those is an important control. It's also used in ecology and in crop improvement and in tissue culture. And you can also use it, as we've shown, to characterize unsuspected phenomena, for example, somatic endoreduplication in Arabidopsis. And we've more recently been using it for molecular and cellular biology of the nucleus, and I'll end up with a few comments about that. In the next slide, I just wanted to emphasize that the chopping technique can be used with animal tissues and organs, as well as other forms of life, insects and nematodes we've chopped up, as well as mammalian species. And in this histogram, you just see a DNA distribution coming out of chopped up brain tissue. And the ease with which you can release nuclei from chopped mammalian tissues, it makes this kind of approach, I think, an important one for studying the molecular biology of those cells. In the next slide, going back to the plant story, we were interested now in extending the genome size observations we've been making to the entire angiosperms, angiosperms being defined as the flowering plants. We estimate, or others, about 500,000 species of flowering plants in the world. And part of the problem of anthropogenic change is that about 15,000 species of all organisms are likely becoming extinct per year. Some of these, of course, will be plant species, and some of these plant species are as yet undescribed. If you want a feel for this, about 2% of known species have a minimal basal molecular description. Of course, the major crops do, but beyond that, there are very few plants that have been systematically studied. And so there really is a need for a global molecular census of the angiosperms before they all disappear. If we go to the next slide, what kind of global inventory should we start with? I think we should start with genome size measurements simply because it's easy to do, and it's fundamental to our understanding of eukaryotic species. For genome sizes, there is a database available in the Royal Botanical Gardens in Kew. The limited information is available there. About 1 to 2% of the flowering plants are categorized there. But as I've mentioned, and I've already shown, flow cytometry is ideal for measurements of C values, and you can extend them, I think, now to all 600,000 species. But one problem you immediately encounter is something called the dynamic range of C values. That is the range over which C value can vary within the plant kingdom. And the next slide from the work reported by Gregory and also Botany just illustrates this problem. And I've circled in red the angiosperms to illustrate the span of C values that were in the literature that he categorized at that time. And you can see they span an enormous range. And as you'll see in a minute, it's even larger now as more values have been made. So this is problematic. It means if you chop up a plant species, you're not quite sure what genome size you're going to encounter if it's unknown. And this is where, in the next slide, the BD Accuri C6 portable flow cytometer comes in. So this has two lasers, it has no user adjustments, a 24-bit ADC, which is an important feature, which implies that it has the dynamic range to cover a large span of fluorescence values, as would be found with the plant nuclear DNA content measurements. And it has no pressurized sheath, so that the inputs that you have for this instrument are minimal. It's essentially portable. The next slide tests the ability of the C6 flow cytometer to analyze the full range of plant DNA contents in the lab. We selected a series of plant species, Arabidopsis thaliana, which has a 2C DNA content of 0.32 picograms, going all the way up to Alstroemeria aurea, lily of the Incas, which has about 81 picograms of DNA per 2C nucleus. And then we took these and we measured them using the C6. And in the next slide, first we established the conditions for using the C6, using Pisum sativum, which is a nice model species, a garden pea. We measured the orange fluorescence, essentially FL2, against red fluorescence. For the homogenates, and you'll see in panel A, you get signal from a lot of cellular debris which is present in the distribution, but you also see this rectangle, which I've outlined in red, of correlated fluorescence, about 1.8% of the population. If you gate on that region, in panel B, you see on a uniparametric histogram now a very nice distribution of DNA contents. And so if you go to the next slide, for Arabidopsis thaliana. Again, if you look in panel A, you see the distribution of red versus orange fluorescence. You see in region R1, which is expanded in panel B to full scale. Again, you can find a region, P1, of correlated fluorescence representing the DNA content. I should mention that the fluorescence that we're measuring here comes from propidium iodide ribonuclease staining, and the reason that we have correlated fluorescence is because we're spilling over the propidium iodide fluorescence into the FL2 and FL3 channels. But being correlated, you could put a window around it, P1 window, and then gating on that you get in panel C a logarithmic distribution showing very nicely the 2C, 4C, 8C, and 16C, and possibly even 32C peaks. So that was encouraging. So on the next slide, we then took the chopping procedure to all of the plant species that I previously mentioned. We chopped them up. We stained with propidium iodide ribonuclease. We, in panel A, measured the orange versus red fluorescence in a dot plot. We took region R2, which encompasses the region where the nuclei would fall, and we identified this line of correlation containing all the nuclear peaks. We put a P2 window around it. And then in panel C, we have a log histogram in which you can see all the various peaks that emerge. There's the Arabidopsis thaliana 2C, 4C, 8C, and 16C peak is not labeled, but you can see it there. Pisum sativum 2C and 4C, and then Triticum aestivum, that's wheat, 2C, and then Alstroemeria aurea, 2C peaks. Now, the positions of all these peaks indicates the amount of DNA that's present, and we also know the amount of DNA that's present from the C-value database at Q. And so panel E then allows you to plot the fluorescence that we measured against the known DNA content. As you can see, it has an extremely high level of correlation. In other words, the propidium iodide fluorescence that is measured by the instrument accurately tells us the amount of DNA that's present. So on the next slide, If we think about spanning the angiosperms, the smallest angiosperm genome size is that of Genlysia margariti, which is a carnivorous plant, has a very small DNA content, 2C value of 0.129 picograms, so that's about a third that of Arabidopsis. The largest currently measured is that of Paris japonica, which has a 2C value of 304 picograms. So you've got an enormous range, but this is still smaller than the dynamic range that's measurable using the Accuri C6. So this implies, in the next slide, that we can consider analyzing all the species that we can find in the world. The throughput and time frame seems appropriate. 500,000 species. If we place 30 machines around the world, run them at 8 samples per hour, 40 hours per week, we could do the 500,000 species in only 52 weeks of total time. And if we wanted replicates, it would take 2 years or 3 years, depending on numbers of replicates. Of course, collection and availability of local taxonomic expertise will be the major issue, and those problems would have to be sorted. But the measurements themselves are not going to be rate limiting. So where does this lead us? On the next slide, availability of the C6 means we're not limited in the technological ability to get a complete description of genome sizes for flowering plants. You can imagine using GPS-enabled cell phones with cameras to link samples and locations, and then, of course, we'd move on to next-gen and Gen3 sequencing to describe all of these genomes starting at low coverage. On the next slide, in the last couple of slides, I'd just like to talk a little bit about flow sorting in plants. The Accuri C6 cannot be used for sorting. It's not a sorter, although Becton Dickinson is now going to be releasing a more or less portable sorter as well. But given the ability that I've shown to make suspensions of nuclei, we can now think about experiments involving sorting. And in particular, we're interested in cell type-specific gene expression. If you go to the next slide, here's just a general schematic of the plant cell in which the nuclear genome produces messenger RNA through transcription, which is then translated into protein. The messenger RNA itself is degraded. And if you consider gene expression on the next slide, generally what is done is to take poly A plus RNA from the whole cell and analyze that. And the sampling of this estimates, of course, the steady state concentration of RNA within the cell rather than its synthesis. And so we've been interested in looking specifically at the nucleus. And in the next slide, we call this approach nuclear transcriptomics, and we have a couple papers that have come out describing this approach, which is also applicable to animal cells, I should mention. And the idea is to label up the nucleus of specific cell types using a fluorescent protein. And in the next slide, you'll see some protoplasts which are expressing a suitable GFP construct, which is a fusion of GFP to the coding sequence of a histone. And when this is expressed within these tobacco protoplasts here, you can see green fluorescence in the nucleus, the red fluorescence representing chloroplasts, and the black areas representing the central vacuole in the cell. In the next slide, you can see what happens when you use promoters that are cell type specific. In this case, we've generated transgenic plants, and we're looking at the roots. And the green fluorescence in the left panel is driven by a sulfate transporter promoter, which is specific to the phloem companion cells. And in the right panel, it's driven by a promoter that's specific to the endodermal cells. And you can see the green fluorescence in the nuclei of these specific cell types. And so what we can do then is simply chop up the plant material and release the nuclei and analyze them. In the next slide, what you see is a biparametric analysis of DNA content versus GFP fluorescence, and you can see the position of the endoreduplicated peaks in the dot plot in the control, which is the upper panel, 2C, 4C, 8C, and 16C nuclei. And then in the lower panel, we're using a plant in which a promoter that is constitutively active is making green fluorescence within all of the nuclei. And you can see a region of the nuclei now which are green fluorescence as well as endoreduplicating. So the C6 is very useful for identification of suitable transgenic plants since it allows very rapid screening of large populations. In the next slide, you can see examples in which we've taken many of these different types of plants and we've chopped them up and analyzed them. And you can see interesting things immediately. So in panel E, we're looking at a promoter which is active in the endodermis, and you can see from the DNA content distribution that those cells are 4C and 8C, whereas for the other cells in the phloem companion cells in panel F, you can see that there are 2C and 4C. So interesting information pops out immediately that using the C6. If we go to the next slide, you can see that, again, I should emphasize C6 is not a sorter, but if you use a sorter, you can then sort out the positive nuclei, and in region R6 in panel B, you can see we sorted these GFP positive nuclei. We amplified transcripts from these sorted nuclei, compared those transcripts to control transcripts using microarrays, and identified the genes whose transcripts were upregulated. We took the top 12 genes of these and found the promoters for those genes and used those for regulating GFP expression in new transgenic plants. And in the next slide, you'll see all 12 of these showed phloem or vascular-specific GFP expression. So the approach of looking at a nucleus to give you the transcripts that are specific to a specific cell type seems to be valid. And the next slide just shows all of these different plants going from NPCC1 to 12, and you can see the patterns of GFP targeting to the vascular and phloem companion cells is maintained. On the next slide, you might reasonably ask, how does this compare to conventional transcriptomics? We have shown in my lab, and I won't go into the details, that transcript levels estimated from nuclei are broadly concordant with those estimated from protoplasts, and our results using nuclei agree with those of other groups using sorted protoplasts. And one of the references is down below is the collaboration with Philip Bentley's lab. In the next slide, we can ask the question, is this approach applicable to animal systems? Flow sorting specific cell types labeled with fluorescent protein expression is, of course, widely used in animal cells. But you can have difficulties when you're dealing with complex tissues and organs in preparing single cells from those tissues. And I mentioned the central nervous system before. Very tricky to get individual cells out of the central nervous system. So it would be appropriate, I think, to sort nuclei, and we'd just use histone GFP fusions to label the nuclei within specific cell types in the brain, and then use the chopping technique to get the nuclei. And the next slide just shows that we can label, in this case, live HeLa cells, the nuclei of these cells, by expressing histone 2B GFP fusions in this case. The final slide here, does the spectrum of RNA transcripts of animal nuclei correlate with that of the cytoplasm? We've done some work in which we looked at HepG2 cells grown in culture, and we looked at and analyzed, in fact, sorted DAPI stained nuclei, and then looked at poly-A plus RNA and microRNAs and correlated the expression profiles between cells and nuclei using microarrays. And as you see on the next slide, the correlation is good, whether you're looking on the left panel at microarrays of transcripts or of microRNA microarrays in the right panel. So in the final slide, I think conclusions are that the nucleus is a valuable source of important information in eukaryotic organisms. The BD Accuri C6 flow cytometer does provide unique features, make it particularly suitable for analyzing nuclei. And I think the type of information that we can obtain using this instrument will be of profound significance in the future of scientific research in this area. And so with that, I'd like to pass this back to Tamlin, and I look forward to answering any questions you might have.
Thank you, David. I want to apologize once again for that difficult start, but I think once we got going, he had a lot of good information there. And if you have any questions for him, please do not hesitate to type them into the Ask a Question box, and he can answer them at the end of the presentations. Our second speaker is Chuck Cannon. Chuck, we're ready for you this time. Associate Professor at Texas Tech University and Professor at Xishuangbanna Tropical Botanical Gardens in Chinese Academy of Sciences. Chuck will discuss the opportunities and challenges inherent in genome sizing of tropical plants and how he has used flow cytometry in his research. Chuck, ready when you are.
Thank you, Tamlin. Today I'm going to be talking about genome sizing of tropical plants, and I think there's a lot of opportunity there because we don't know that much, and there aren't as many challenges as there used to be because of the many technological advances that have been happening. For example, the BD Accuri system that we're talking about today. Next slide. And we all know that tropical forests are very vital to the global economy and ecology as well. And we've been hearing for many years about Save the Rainforest campaigns and trying to cut back on consumption of the rainforest for various things, but we actually still know very little about the rainforest. Many basic things we don't have, like genome size. And one of the challenges is there are so many species. There can be thousands of species in a single forest. Next slide. So the research questions that we've been interested in addressing are fairly basic. For example, the first one, what is the typical range of genome sizes in tropical tree species? Like this kind of thing is not very well known, because the question of why are there so many species in tropical forests is a big one that we don't really have a good understanding yet about. So the second question is, does genome size evolution play a major role in the diversification of tropical tree species? This genome size evolution does play a major role in many herbaceous plants, and we want to know if that same thing is happening in tropical species. Are there consistent differences between temperate and tropical trees? So there are many gradients that we see going from the tropics to the temperate zone. A decline in species number, a decline in certain other aspects, and do we see a consistent difference between temperate and tropical areas? And does fractional differences in genome sizes play a major role instead of this kind of whole ploidy level that we see in some herbaceous groups? Next slide. So just a few things that we do know about plant genome sizes. Even though we've been able to measure genome sizes for over 50 years now, still we know less than 1% of angiosperm species. We don't know their genome sizes. So you can see at the top graph there's a very wide range in angiosperm genome sizes, and if you look at the figures A and B, they're showing angiosperms and gymnosperms. Although there's a few examples of very large genome sizes in angiosperms, most of them are much smaller than the typical gymnosperm. And so you might have heard, stories about the massive genomes that pines have and things, but we don't have as much of that in angiosperms or flowering plants as you do in gymnosperms. But still, we don't really know that much about genome size in general. Next slide. There are a few things that are known. Typically, tropical herbs do seem to have smaller genomes, but this hasn't been studied very well at the family level or in trees. Within a few families, it's been shown that temperate species have larger DNA amounts than tropical ones, so there's a little bit of conflicting evidence there. And in general, trees are predicted to have smaller genome sizes than herbaceous plants. Next slide. And so to address these questions, we chose to start with the Fagaceae. This is the oak or the beech family, so it's a worldwide plant group. It's not in Africa, actually, but there are 8 genera, and it's all trees. There are about 1,000 species around the world, and you can see from this graph that there is a geographic pattern as well where you have several genera like Notholithocarpus or Cycolepis in North America. And so there's a major center of diversity in North America with Quercus, which is the oaks, and chestnuts, which is Castanea, and that's the blue circle there. And if you pay attention to the color coding here, it's consistent through the presentation where the blue things are temperate and the pink things are tropical. And so if you look at the tropical areas over there in tropical Asia, where most of the tropical Fagaceae are found, there are a couple of genera that are there, Lithocarpus and Castanopsis, that are not found anywhere else. This is a major center of diversity. So it's a very good group to study because you have a nice contrast between temperate and tropical areas, and there's a lot of diversification that has happened in both places. And so it's a good group to study. I mean, the temperate oaks are fairly well known. They're one of the better studied tree groups, and so we can do some nice comparisons there. Next slide. And just looking at the phylogeny and the species richness of the family, again, the color coding is the same. And so you can see, there has been a major diversification with Quercus, which is the oaks, 450 species worldwide. And it's purple, so it's present both in the temperate and tropical zone. But then you have Lithocarpus, which is also a major diversification. In tropical Asia only, you have 300 species. And then you have some smaller genera there, Notholithocarpus and Chrysolepis, that are just relictual things found in the Pacific Northwest, and a few of these odd things, Trigonobalanus, Columbobalanus, that are found in the tropics, but you only have one species for each genus. And in general, the ploidy number is fixed for the genera, so we don't have a lot of evidence that there's polyploidization or whole genome duplication going on. But again, very few individuals have actually been measured, and so that was part of the point of the study. Next slide. So the method that we chose, using the next generation flow cytometry that's offered by BD Accuri, the C6 system. It's a very nice compact system, and we got it up and running fairly quickly. The buffer system we're using was David Galbraith's buffer. The previous speaker established that. We used the propidium iodide for the fluorochrome, and we had an eternal standard of the garden pea, which is a standard thing to use when you're measuring genome size with flow cytometry. And we've explored a range of tissue types for extracting the samples, and from adult leaves all the way over to young roots and shoot. You can see in the little diagram there showing the tubes, just the darkness of the liquid indicates, that there's a lot of things when you extract from the leaves, you get a lot of other compounds that are along with the nuclei, and so it's a more difficult sample to accurately size. And we found definitely that root tips were much better, and so we were actually germinating seeds and taking the young root tips, and those were giving us very nice results. Next slide. And so this is an example of 4 different species from 4 different genera, so A, B, C, D. So the A peak on the first graph is showing you the nuclei that we measured coming from Lithocarpus elizabetha, and that's compared to the internal standard, which is the P. And so that's why we have the internal standard in there to compare with the thing that we are measuring so that we can adjust any kind of, differences in that particular sample run. And so there's an internal standard in every one of them, and you can see the P is very sharp and clean. In some of the others, there's other material coming in with these samples, and so you get a bit of scatter, but there's a very clear peak. And so you can very clearly and objectively measure these genome sizes for these 4 different species and 4 different genera. So the machine performed very well. We were very happy with this. Next slide. So the data that we did collect, again, we focused on the tropical Fagaceae. So we collected 40 new species for the family. There had been, you can see in the right column there, the literature data, there were 32 species previously known, heavily concentrated in temperate Quercus. And so we measured 40 more species, and this is including 147 individuals. We were sampling multiple individuals of many species. And all of these measurements were new accessions, and this work was all part of Ms. Chen Sichong's master's dissertation research in my lab at Sichuan-Bana Tropical Botanic Garden. Next slide. So some of the results that we found, that it does seem that genome sizes are slightly larger in tropical species, and this is independent of the history. So one of the reasons to look at this in a phylogenetic context is you can understand the evolution much better. And you can see the phylogeny is on the left side and the sizes are on the right side, and so you have these box and frame graphs showing you the mean. So in the middle of that box is showing you the mean for each one of the genera for the various species that we measured. And you can see the Lithocarpus and Castanopsis are larger, even though they're independent diversifications that happened in the tropics. And so it seems that there is this association between smaller genome size in the temperate zone and slightly larger genome size in the tropics. Next slide. And so this was very interesting. If you actually look at the Quercus, as I mentioned earlier, there are tropical Quercus and there are temperate Quercus. And so you can actually look at these genome sizes as well, and the same pattern is present at the genus level. And so the subgenus Cyclobalanopsis appears to have slightly larger genomes than other subgenus, which is Quercus. And so you do have, again, the same pattern where the tropical species have slightly larger genome sizes. Next slide. So the basic results, just to kind of sum up some of this, is the overall variation at the family level is relatively small. You only have 2x from the smallest to the largest, and this is relatively consistent with the idea that the whole genome duplication and polyploidization hasn't really played a major role in the diversification of these tropical species. And so that's quite interesting. I mean, this was kind of one of the assumptions, that this wasn't happening in these tropical trees, but we needed to prove it, and we didn't really find any examples of obvious polyploids, even though it has been observed in a few tropical species, but we didn't directly observe any polyploids. And so from this, it definitely seems that some kind of mechanism other than polyploidization is driving this diversification of things. We do see some within genus variation, which is quite interesting, but relatively little intra-specific variation. So when you're measuring numerous individuals of one species, they all pretty much have the same genome size. And that's, again, a testament to the accuracy of the machine and how you can use it very effectively. You get the same reading from the same species most of the time. But still, there is some within genus variation that existing within the genus. So the different species within one genus do have slightly different genome sizes, up to about 20-25%. That then leads us to better questions. And so we didn't know anything about genome size before, and now we can begin to ask more detailed questions about, well, what really is going on within the genus, and what are these slight differences in genome size doing to the genetic diversity or the phenotypic diversity in that species. Next slide. So tropical species do seem to have larger genome sizes, and even though it's hard to determine the direction of the evolution from the phylogeny, because it's still a relatively simple phylogeny, you don't have a lot of shifts between the temperate and tropic zone, you only have a couple of examples. So it's a little bit hard to tell the direction of the evolution, but it seems, particularly looking at the Quercus example, if you remember, the Cyclobalanopsis was kind of basal in the genus, and so that was larger. It seems that the deciduous or the temperate species have reduced genome sizes, so it seems that genome sizes are getting smaller as things diversify in the temperate zone. And this is relatively inconclusive, but that seems to be the pattern. And so this pattern is true both among the genera in the family and then also within the subgenera in the genus Quercus. And like I mentioned on the previous slide, that the species within a genus can vary up to 20-25% in their genome sizes, and so some kind of, contraction and expansion of the genome is occurring, and that's maybe driving the genetic diversity that you find in the tropical species. And one thing that was kind of an assumption was that tropical tree species have relatively small genomes, but again, you don't know that until you really prove it. And so we found that, yes, it's true that tropical trees don't have massive genomes like pines or other infamous plants like Trillium. So, this is good news for further genomic studies. And so we are now confident to move on to begin to sequence a lot of these things. And from looking at the previous point where, we have one species that's 25% larger genome size than others, you can actually begin to choose which species might be interesting to sequence. And so this is providing us a lot of rich information for improving our sampling design, for improving our questions that we're asking. So it's very basic information, but it's actually very critical for improving the research and improving our knowledge. Next slide. We're doing a little bit of study in the family Juglandaceae. This is the hickory or pecan or walnut family, and you can see in the little phylogeny there the red box around Fagaceae and the red box around Juglandaceae. So they're fairly closely related. They're in the same order. They diversified maybe 50 million years ago, and they also have some examples in the temperate zone and in the tropics. They're not as diverse. There's only a handful of species in each one of these areas, but we were wanting to look at this family as well to begin to see if there was a broader pattern where you actually do have this consistent difference between temperate species and tropical species. And so far, we've measured 5 species, and 2 were in the database already, and so we now have a total of 7 species. So it's not an overwhelming amount of evidence, but it does seem, again, that the tropical species, which are represented in this genus Engelhardia, they do seem to have slightly larger genome sizes. So this is good, additional support in another family. Next slide. So the advantages of the BD Accuri machine that we found so far is that, the small footprint and the self-contained system easily allow us to move it around from location to location. So we're working in a botanic garden. It's quite a remote place. It's an hour drive from the nearest airport, which is an hour flight from the nearest major city. So it's very convenient to be able to take this machine to this botanic garden because, as I mentioned, the tissue is very critical. So getting good, fresh samples is really a limiting step. And so when you can go out and collect the seed from the plants, or even the leaves do work, particularly when they're fresh. And so if you can go out and get the good sample right away from the plant, you can overcome a lot of the challenges that you're going to have in preparing samples and getting accurate measurements. And so being able to take the machine to the forest is actually a major advantage. You know, it has very simple instrumentation. It has this wide dynamic range that Dr. Galbraith talked about, and it allows you to compare many different plants without any kind of recalibration. And so, you know, our study right now is limited to a couple of families of trees, but because we're in a botanic garden, it would be very easy to expand the study to a wide range of species, from palms and many different kinds of plants. And so we could be able to do that without having to change the machine much. And actually my student, Ms. Chen, is now in Kunming, and she's now setting up the machine to work with a seed bank. And so seed banks typically go through germination trials. So they take the seeds out and they germinate a few and make sure that the seeds are still viable and they're being able to store them properly. And so when they do this, they would just throw the seedlings away. But now they can take the seedlings and get genome-size measurements for everything that they're germinating in their trials. And so it's actually a very nice thing. They can sit there and they can gather a lot of data that was just being thrown away previously. And so it's a very nice thing. And so we've also been able to get very consistent results in a relatively short period of time after training with the machine. Ms. Chen didn't have a background in this kind of work. I mean, she was a good lab person, but she didn't have any experience on these type of machines before. She very quickly learned how to use it and was able to get results within a short period of time. Next slide. So some of the challenges that we do face, just to mention some of these, is that in the tropics, it's a very wet, humid climate, very warm, and there are many living things everywhere. There's a lot of things in the air. So anytime you're studying using optics, it can be difficult. I remember studying primates, and I had binoculars, and my binoculars would grow fungus after a few months. And so anytime you're doing something like this for long term, and so we keep the machine there for over a year now, and things begin to grow on the surfaces, and so you have to be extra careful to try to keep the machine dry, and so you really need to keep it in some kind of special lab conditions. So say there's a second point there that, you know, the air is really full of life. There's lots of spore. Things can infect the buffers in the tanks, and so we've had buffers turn green. So you really do have to take a few extra precautions to make sure the machine is clean and protected. And if you can put it in a special room where other people are not coming in and out, we've actually had some problem with people working with soils, and the spores were coming out of the soil samples and things and infecting our machine. And again, the selection of material is very critical. Leaves are full of secondary compounds that inhibit the labeling process. And so if you can get these root tips, it can help a great deal. The chopping is very important, and that can take some time to perfect. And again, that depends on the material you're using. Whether you're using mature leaves or root tips, your chopping style has to change slightly, and you just have to be patient with that. That was the most challenging thing that Ms. Chen faced in her studies. She did have to optimize the buffer a little bit as well, and so, you know, different plants have different compounds in them, and so I would expect as you sample more plant species that you'll encounter situations where you're going to have to optimize the buffer in some way. It wasn't a great deal of optimization, but she found that adding a few things to the buffer was very helpful in getting good, consistent results. And I think that's pretty much it, and so I'll go back to you, Tammy. Thank you very much.
Thank you, Chuck. If you have any questions for Chuck, please submit them now by typing your question into the Ask a Question box on the lower left of your screen and hitting Submit. Our third and final speaker is Claire Rogers, Senior Marketing Applications Specialist at BD Biosciences Cell Analysis. Claire will talk specifically about the BD Accuri C6 flow cytometer, providing details on the attributes and its functionalities for DNA analysis. Claire, we're ready when you are.
Okay, thank you so much, Tamlin. Now that you have seen some of the interesting applications being done out there in the field with the Accuri C6, I just want to give you a brief overview of the system, point out some of the ease of use attributes, specifically relative to DNA analysis for those of you interested in this sort of application, and mention a couple of QC tools available through BD Biosciences, particularly for DNA analysis, and also mention ways that you can obtain cell cycle analysis from your C6 data. Next slide, please. Here are photos of the BD Accuri C6 flow cytometer system. On the left, you can see the C6 in its single tube feed format, and you can see how small it is in terms of footprint, even including the fluidic bottles on the left there, as compared to the monitor and computer that come with the system when you order the system. And on the far right, you can see a C6 on which has been mounted the C-sampler automated handling arm. Even if you purchase a C6 in the single tube feed mode, you can always opt to later purchase the C-sampler arm to add on to it if you so desire. And from the sampling arm, you can see in this photo a 96-well plate is loaded up there, but it can also handle 48-well plates and a tube rack for 24-well tube rack for holding 12 by 75 millimeter tubes. Next slide, please. The C6 system is innovative in a number of ways. In fact, all of the major components that make up any flow cytometer have innovations in the C6 system. And I'm going to take you on just a quick tour of some of those, hit some of the highlights on the fluidics, the optics, signal detection, and the software. And I believe there's time for question and answers at the end of this in case you need clarification on anything. Next slide, please. The fluidic system is quite unique. It's a non-pressurized system, but it is not a syringe-driven system as are so many small cytometers or benchtop cytometers these days. It is a peristaltic pump-driven system. So the peristaltic pumps, of course, have a push-pull action in them, and our lead engineer has developed a patented pulse dampener that smooths that push-pull flow out and enables the system to create a laminar flow fluidic. So it's able to take advantage of the hydrodynamic focusing that one has with laminar flow fluidics. And this allows also very fine control by the user over the flow rate and the core size so that you can flow your cells slowly past the laser beam, which is quite important in getting nice DNA cell cycle analysis histograms. It also enables the system to know how much volume has been pulled from each tube so that absolute counts can be determined without having to add a counting bead to your sample. In addition, any kind of tube that you can fit onto the sample introduction probe can be used because no seal is formed since it's a non-pressurized system. So you can use 12 by 75 millimeter tubes of all plastic types. That doesn't matter. You don't have to buy a particular tube. You can also use conical Eppendorf tubes, 1.5 mil size or even 0.5 mil size if you have very small sample volumes. You can go down as low as 50 microliters in the bottom of a conical tube. The system can go up to speeds of 10,000 events per second if desired. That would not be typical for DNA or cell cycle analysis, but one can do that. The picture you're seeing there on the left is really just a cutaway engineered schematic of our sample delivery and flow cell system. And the sample actually is pulled up from the tube, which would be at the bottom of where you see the word sample, would be below that point. The cells are pulled up. The laminar flow is created, so the cells are flowing up past the laser beam and then off to waste. And there are controls on the system in order to clear bubbles and to do back flushes and to clean the flow cell, et cetera, all of the controls that one would expect to have. Next slide, please. This is a cutaway picture of the optical bench of the system, and it is very small, very compact. There are virtually no dichroics that come between any of the fluorescence or light scatter detectors and the flow cell, and this improves signal detection quite a bit. The system comes with two lasers, a 488 nanometer blue laser and a 640 nanometer red laser. It has 4 fluorescence detectors. Those are photomultiplier tubes. You can see they're labeled with colored labels FL1, 2, 3, and 4 around the top. And there is a forward and side scatter pin diodes for light scatter detection. optical filters that come standard in the system are listed there on the labels I've placed on top of the photomultiplier tube. So they're optimized basically for FITC, PE, PerCP, Sci 5.5 for FL3. And FL1, 2, and 3 are going to read fluorescences due to blue laser excitation. FL4 is dedicated to reading emissions due to red laser excitation and is the standard filter that comes with this optimized for APC detection. But there are optional optical filters that are available. Those are listed over on the right-hand side, and so for specific applications, one might want to choose one of those optical filters instead of one of the standards. There is another upgrade called the selectable lasers upgrade. The standard configuration is what we call the 3 blue, 1 red. In other words, FL1, 2, and 3 are reading blue laser emissions, and FL4 is reading a red laser emission. That's 3 detectors from blue laser, 1 from red laser. The selectable lasers upgrade is just a simple software upgrade that allows you to then switch to operating in a 2 blue detectors, 2 red laser detectors, or you can operate in a 4 blue laser excited detection mode, which essentially turns off the red laser. So this gives you more flexibility in terms of fluorochrome selection. Now, the other unique feature of our system is the fact that the signal, not only is the alignment of the lasers and the optics optimized and locked down at manufacturing, but the signal detection is optimized for you as well. In other words, the voltages on each of the detectors are preset to give the optimal fluorescence detection allowable for the system. And we have added an extra log of dynamic range to our fluorescence detection range so that you can see very dim and very bright signals all at the same time, so that obviates the need for voltage control. Next slide, please. One of the advantages of the pre-optimization of the system is it greatly simplifies instrument setup, and talk about that in just a minute for the individual user, but it also means that instrument-to-instrument variation is very minimal. So what you're looking at here are overlays of the 8-peak setup beads that we use in manufacturing and that we recommend our customers to use every day when they validate performance of their instruments. And these overlays that you're looking at were collected from multiple C6 instruments over a 6-month period as they were manufactured at our site in Ann Arbor, Michigan. And so what this is telling you is that the voltages that are applied to the detectors are very similar, give similar performance for fluorescence detection from instrument to instrument. So if you are trying to do cross-comparative studies, the C6 is a great instrument for that, so long as both sites are keeping their instrument clean and they are doing their bead qualification every day to be sure that the instrument is still in specification. Next slide, please. So what this means is that setting up your instrument is hugely simplified. Essentially, once you have your instrument set up, you're going to power it on each day. It takes about 15 minutes for it to come up to full warmed-up power on. And then you will run a tube of water, a tube of diluted hypochlorite solution, and another tube of water, and this helps clean out the SIP and the flow cell. And then you will validate the performance of your system with beads, the same beads that we use in manufacturing to set up the system. You'll use those beads to validate the performance of your system. And once that validation is performed, you are essentially ready to go. You don't have to make any decisions about setting voltages or anything else on your system. So it's quite simple to get up and running. Next slide, please. The software is also innovative. And here's a screenshot of our manual collect tab in the D-Flow software. The company we work with, Menlo Innovations, has people there called high-tech anthropologists, and they don't write code. They don't operate as computer scientists exactly, but they analyze how people interact with software. So in the process of developing software for the C6, the high-tech anthropologists worked with both advanced cytometry users and beginner users, watching them work with current flow cytometry softwares of various types to see where the pain points were, And then as they developed the software plans and layouts for our system, put them in front of those same users and had them try it out and say, so is this working for you? Is this not working for you? And in that iterative process, we were able to come up with software that is very intuitive. It has all of the high-end accessibility and options that people want in terms of being able to create multiple types of gates. They have event coloring, et cetera. But also, it's easy to start working with. Most of the controls are right up front. They're not buried in menus, et cetera. That was an innovation, and we always get high praise for our software, especially for new users. It's very intuitive and simple. Next slide, please. Another interesting and useful tool for some researchers is the fact that the instrument is quite portable and very robust. And here's a few photographs of the C6 out in the field in action. Over on the left, it's on the back of a small boat. We took it out on Lake Erie, and we're sampling water, looking for toxic algal blooms with some researchers from NOAA. And in the upper right, you'll see the C6 there, and there's a black strap around the middle. So this is on a ship that's out in cruising around the Antarctic and taking water samples where the seas can get quite rough, and so they actually strap it down to the bench, and they take it out about a couple times a year on a boat there and do the sampling right where the water is, as opposed to having to store the samples and analyze them later. And there on the bottom you can see one of our European technical applications people, Kate, on the left, and she has the C6 plugged in and working in a car to the car battery. And next slide, please. So some of the functionalities that make the C6 perfect for DNA analysis is that, again, you can control the particle rate. So you can adjust the fluidics rate, and you can slow it down appropriately for the analysis that you need. You can also control the core size for small or large particles, which also helps you get better resolution on your DNA histograms. You can threshold on any parameter, light scatter or fluorescence, and you can assign a primary or a secondary threshold. Many people doing DNA analysis would prefer to threshold on a fluorescent signal to help eliminate debris and aggregates. Speaking of aggregates, again, you can use the area versus height signals on any of the parameters to exclude aggregates. The area and height signals are collected all the time on all signals. So you do not have to worry if the gating wasn't done properly initially. You can regate, redisplay the data, and the data is always there. The instrument has a linear response across its broad dynamic range, which actually was very well illustrated by Dr. Galbraith's data showing the wide range of DNA intensities that was able to be collected in a linear manner. And there's another tool called the virtual gain tool, which allows one to use an internal standard in DNA staining to compensate for sample-to-sample staining variation. So many people like to add, for instance, something, if they're doing a mammalian cell cycle analysis, they may add trout erythroid cells or nuclei to each sample, so it has a very different DNA content, will show up in a different portion of the plot, and those cells get stained along with the sample and then run on the flow cytometer. And typically people will adjust the voltage on the fluorescence detection parameter for the DNA stain, adjust the voltage to place the internal standard in the same median channel value for each sample collected. And this allows to compensate for the fact that there are going to be slight differences in staining of the DNA between tubes. With the C6 system, you collect all of the data up front, and then you can use what's called our virtual gain to align all of the data using your internal standard to a user-determined channel. That's a very nice tool. Next slide, please. The BD DNA QC Particles Kit contains two different kinds of particles, which can be helpful for validating instrument performance. One of these are chickerythroid nuclei, which tend to form aggregates. These chickerythroid nuclei signals can be used in two ways. First of all, the CV of the first singlet peak that you see there on the far left should be less than 3% if your instrument is clean and aligned well and running properly. So you can see in this instance we have a very nice CV on the first peak of 2.5%. And these cells tend to form aggregates, and so it allows you to look at the doublet, triplet, quadruplet, et cetera peak, see where they fall and check that your instrument is linear. Next slide, please. The other particle included in the kit are calf thymus nuclei, and these actually have a small cycling population. This is a nice way to validate that your instrument is, that you're able to do singlet gating properly. If you look at the left-hand side, you can see the area versus height signals plotted against each other for propydium iodide stained CTN, and P1 is encompassing the singlet populations. Aggregates will have a different proportionality between the area and the height signals than the singlets have, and it allows one to identify them and gate on them. And then the histogram plot to the right is gated on just the singlets. And again, there's the percentage of cells in the G0, G1, and G2N peak of the CTN is qualified and written on the particular packet of QC particles that is purchased, so that you can be sure that your staining, your analysis, and your cell cycle analysis algorithm are actually functioning properly and returning the proper percentage of cells in G0, G1, S, and G2M phases. Next slide, please. Lastly, I just want to mention in terms of cell cycle analysis algorithms, there are a wide variety of algorithms out there, and I am not expert enough to recommend which one to use, but I can say that our BD Accuri FCS files created in our software can be exported and analyzed in multiple other-party software analysis programs. So on the left in this plot, you can see an example of some Accuri data as it appeared on the C6 system. This was a cell line stained with propydium iodide, and it had a fairly decently high S-phase population. You can see that's the bump in the middle there between the far left high peak and the right-hand third peak. And this data was run through FCS Express using multi-cycle. So one can purchase FCS Express with multi-cycle as an add-on. And if you do that, you can run through those. They have several popular algorithms in the multi-cycle software that can be used. FCS Express is nice because it has an Accuri file importer that can be purchased along with it, which really simplifies pulling data into the analysis program. It essentially goes into your C6 file and grabs the FCS file for you, and also can grab the plots and gating strategies that you used and then recapitulate those in the FCS software. So that's a great time saver there. FlowJo software by TreeStar also can handle and analyze our data files, as well as ModFit LT, which is produced by Verity House. All of these companies have worked with us, and we keep in touch with them and send them our data periodically to make sure that we are maintaining compatibility with their programs. And that is the end of my presentation. So back to you, Tamlin.
Thank you very much, Claire. Before we begin the Q&A session, I want to encourage you to submit questions one final time. We have a lot of questions, and we're going to answer as many as we can. I have a bunch of questions for David first. David, the first one's an easy one. What is CV?
Okay, CV is just the normalized standard deviation, and most of the instruments, flow cytometers, will compute this for you automatically. So it's the standard deviation of the measurement divided by the mean.
Okay. The next question is actually a multi-part question and sort of two questions combined, so I'll ask a bunch of them. What types of DNA fluorochrome are recommended in plant genome sizing? Are there large differences between relative AT and GC content of these plant genomes? If so, is it important to use something like propydium iodide rather than AT base pair specific HOCHT 33342?
Yeah, this is a question from Jim Leary, who's an old friend of mine. It's certainly important to use something that intercalates like propydium iodide for these measurements than a DNA fluorochrome that's base pair specific. Now, other groups around the world, I think in Portugal, have used differences in fluorescence emission from AT and GC regions by different fluorochromes as a way to measure AT to GC ratios in different plant species. And there are a few papers that have emerged on that. But in any case, going back, use something like propydium iodide with ribonuclease for these types of overall genome size measurements.
Okay. Do you need to change the chopping solution for insects, et cetera?
We actually tried this in Chuck's lab. We chopped up some insects that they collected from the botanical garden, and we used the standard chopping solution. So no, you can just use it. In general, I think that insects and animal cells are less demanding than plants for the reasons that Chuck mentioned regarding secondary products produced by plants. So you can use the standard chopping solution.
Okay, and then you measured all species simultaneously?
We chopped separately and mixed and then measured them all together. In principle, you can mix them together and chop. And in fact, people often use this as a ploidy screening tool. For example, if you're looking for a minor population of triploids in an overall population of diploids, you can take, say, 100 seedlings and chop them all together and then look for the triploid peak in the background of the diploids. And so you can make samples and chop them simultaneously.
Okay, here's another question. Have you ever worked with any of the vibrant dye cycle dyes from Life Technologies and Butrogen, and would these be similar to the piRNA dye that you used for your studies?
The answer there is no, we haven't, but they sound to be interesting dyes.
Okay. All right, here's another one. You have mentioned about filtering issues in mammalian cells. Doesn't the nylon mesh help in filtering? Can you suggest any specific and easy-to-use mesh filters for obtaining single cells?
Yeah, I just checked online to see. In fact, you can actually buy these nylon meshes from Amazon.com. I'm not sure we're supposed to give commercials for things, but Nitex meshes. The Nitex is the type of mesh that you can buy in big rolls, and so we would buy a big roll of this and then start cutting it up with scissors and putting it on a holder. So it becomes very cheap.
Okay, someone wants to get some of your expertise on fixation, pros and cons. It seems I can get much more nuclei with fixed tissue.
Yeah, I'd be very careful with the idea of fixation, because then you need to fix the controls to make sure that you're not perturbing the nuclei in any way. But that being said, you can certainly try it and see what happens, and compare unfixed to fixed and do a lot of standard experiments, and it may be helpful for you in certain circumstances. But most of what we do is with unfixed tissues.
All right, Chuck, I have some questions for you now. The first one is, what is the maximum rate that samples can be processed?
Yeah, I think that really depends a lot on the material that you have. I mean, so in our case, we're actually limited by the obtaining the material. So that's the limiting step in our study. But I think, I mean, you could do, I mean, the student, I know when she had all the material there together, she was easily able to do like 12 or 15 a day. And that's being pretty organized and kind of really keeping things moving. But there is the staining step. And so there are a few things that you know, the sample doesn't immediately go into the machine. But you can process quite a few. And I think David even had some statement. I don't remember what it is. David, how much did you estimate you could do in a week or something?
Well, in principle, you could run 8 an hour, but that would be really cranking through it.
Yeah.
And you need to have several people doing the chopping and then feeding the samples to the machine. So the machine can process one sample every 5 minutes or something like that. But you need to have a pipeline to generate all of that.
Yeah.
So realistically, it's at a lower level. On the other hand, I mean, if you're doing ploidy screening in thousands of individuals of a single species, then there's no reason that you couldn't crank through. But if you're doing different species, then you're going to run into all sorts of issues that will need resolution, not least of which is the problems that you encountered with secondary products.
All right, Chuck, do you think the chopping process can be automated?
Again, I think that depends a lot on your question. So if you're doing something like in a crop species or something, I think you could get a very standardized material that you're using and get a very standardized way of dealing with that material. And you could possibly automate it, but I think my student found that was one of the key steps in actually kind of doing a more coarse filtering, chopping instead of the really getting fine material. So I think it's almost like getting the feel for it. And if you're using root tips sometimes and you're using leaf material some other times, so kind of standardizing that is rather difficult. So if you're doing some kind of diversity study, I think it would be difficult to automate that because you're going to have to kind of refine your technique with each one. So I think that might be difficult, but if you're in a kind of more of a crop kind of more industrial setting, you could probably do that.
Yeah, I'd like to add to that. There is a paper in Cytometry last year from an Australian group in which they described automation using a bead beating approach, I think, to homogenize the tissues, but they were working within a single species and a single tissue type. So it can be adapted in that way, but Chuck is exactly right that different specimens, different samples will require personal optimization.
Okay, thank you. Is precipitating and washing the chopped materials after filtration important? Chuck, do you want to answer that?
We didn't find that. I mean, it wasn't necessary for our samples, and I mean, this is my main experience with this technique, and I imagine David could answer that more with more experience.
Yeah, I avoid the idea of spinning things down and resuspending, mainly because every time you manipulate the nuclei, they either tend to break, stick together, or stick to the side of the tube. So just filtering and then just running the homogenate through without manipulation after staining is, I think, the best way to go.
All right, David, this question is for you as well. The Accuri cytometer seems to work well, but I'm afraid we can have a problem if we include in a sample any species with staining inhibitors in the cytosol. Do you try how the measurements using Accuri would work for these difficult species?
Yeah, this is from another old friend, Elvira Slavinska, and she has a lot of experience in this area. And also Chuck has mentioned, dealing with problematic species in his surveys. We should emphasize that there are lots of secondary products that are produced by plant species, mainly to ward off insect attack over evolutionary time, and including various additives routinely in the chopping buffer is sometimes absolutely necessary. And the additives can be things like ascorbic acid, they can be reducing agents, they can be things like polyethylene glycol or polyvinyl pyrrolidone, which are molecules that are designed to absorb phenolics and other secondary products. And so you will have to optimize for difficult species the chopping buffer as well. As a general rule, if the sample changes color when you chop it, turns brown, then that's a bad sign. You've got some phenolics that are being produced or other products, and you'll need to start worrying about that. You can check the effect of these products by, for example, taking chicken red blood cells and taking the supernatant from a chopped plant material that you think has suspicious compounds in it, and adding that to the chicken red blood cells and seeing if it makes any difference to the amount of fluorescence. So there are these little kinds of controls that you can do. You can mix a standard plant, for which you know the genome size, with the problematic plant and co-chop them and then see if the peak positions change. Standard controlled experiments of this type are very useful.
All right, Claire, we have some questions for you now, equipment specific. Are the fluorescence emission filters standard 25 millimeter filters so that different filters can be put into the device by the user?
So different filters can be put into the device by the user, but they do have to come from Accuri cytometers. So they're not totally standard. It's nothing that you can call up Omega and order. But if there was a particular filter that we don't carry, we do have optional filters. I think they were listed. They were listed on the one of the presentation slides that showed the optic bench. You can see which filters are available. And if there is something specialty that you want, you would need to call Accuri and talk with tech support about the possibility of ordering one to be made.
Okay. Claire, someone wonders if you have to validate before every run.
Not every tube run, but before each experiment, I would validate that the instrument is giving good performance. So both with the 8 and 6 peak beads that come with the instrument, and then if you're doing DNA analysis, you would most likely want to validate with some sort of control sample that either you produced yourself from the type of cells that you're working with, or with possibly one of the standards like the CEN or CTN from the DNA QC particles kit that BD carries.
All right, Claire, I have a multi-part question for you. I'm working with protoplasts that range from 20 to 60 microns in diameter. What is the practical limitation of size for use with the Accuri C6 cytometer? Are large diameter flow cells available for this instrument?
So the size of the flow cell is 200 microns in diameter. with the adjustable fluidics, you can adjust the core of the fluid stream to 100 microns. And we have successfully flowed algae species that are from 100 to 150 microns through the system and not clog the system. So you can run large particles through. The one question is whether or not you would get light scatter signals that would be on scale. If you needed to use light scatter to gate, that might be a problem. But if you're doing DNA analysis and you can trigger on, you don't really need the light scatter to gate because you're going to gate on fluorescence positivity, then it may not matter. So a 60-micron protoplast flowing through the system would not be a problem. It would only be a problem if light scatter signals needed to be on scale. Those would undoubtedly be off scale on the standard C6.
All right, Claire, I have another question for you. Are the Accuri list mode data files FCS 2.0 or FCS 3.0 standard file format?
They're FCS 3.0 only. So.
Okay. David, actually, I have another question for you. Can one save protoplast fusion products aseptically for further culture after testing DNA content on the flow cytometer?
Oh, yeah, this is a question from another a former colleague of mine, Dharam Sharma. I reiterate that the Accuri is not a cell sorter, so you can't sort with it, so you wouldn't be able to sort protoplast fusion products. However, if you could sort, or if you were using a sorter, then the question is, can you measure DNA content in a living plant cell? And this is something that I don't think has yet been achieved. There are a number of reasons for this. First, there's the error of positioning of the nucleus within the flow cell, which would increase the CV, which I already mentioned with fixed cells. But with living cells, then you've got to get the fluorescent dye into the protoplast. And at the moment, I don't think people have been able to find dyes that do that particularly well. We've occasionally tried using some of the supposedly viable DNA stains with protoplasts, but we haven't come across anything that works particularly well at this point. But it's an interesting idea, and it would be worth pursuing.
Okay, thanks, David, and thanks everyone for the great questions. This webinar will be archived for 6 months on our website, www.GENNews.com. If you missed parts of it, you can watch it again, or you can recommend it to friends and colleagues. Once again, I want to thank David, Chuck, and Claire, as well as our sponsor, BD. I will be sending out a survey shortly and hope you'll take the time to respond. Your comments will help us to continue to provide topical and timely webinars. Thank you.